This is TWIS. This Week in Science, episode number 825, recorded on Wednesday, May 19th, 2021. How to use your memory with science. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your heads with memory, iconic sounds, and googly-eyed deterrents. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Know your enemy. You may not think you have enemies, being that you are such a nice person and all, but they don't see it that way. No, they see you as the enemy. And by default, that means you have an enemy. How do you defend yourself from this fiend? First, you must know your enemy. You will know them by their words, chosen to influence, not inform. You will know them by their actions, spreading fear, not facts. You will know them by the constant deflections away from knowledge, away from truth, away from reason, empathy, and compassion, and away from the most important strength of our sentient modern minds, our knowledge of reality. The only thing now standing between you and the enemy, the only thing that can preserve your freedom, your intellect, your PIN number, and protect the health and safety of your family is This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. 825. Whew.
Wow.
That's quite a several. We've been bringing the science. We'll continue to bring the science every week. And tonight on the show, we have some fabulous stories lined up. I've got stories about iconic sounds, repairing hearing, and thumb brains. Thumb brains. What did you bring, Justin?
I've got In Search of Life on Other Planets, Teachers Don't Care If You're Smart, Why Horses Are a Thing You've Heard Of, and How to Not Forget Things.
I don't want to forget things. I want to remember things. This is going to be the TWIS's helpful handy tips in the Justin section of the show. Blair, what's in the animal corner?
I have those googly-eyed deterrents you mentioned. I also have two new ways of breathing, one involving bubbles and one involving your rear.
Yeah, we'll get to
my
that.
Ear? Did you say my ear?
Sure.
Well, posterior to that,
Yes.
I need to tell everybody that this is This Week in Science, and if you are not yet subscribed to the show, you can find us all places that podcasts are found. Look for This Week in Science. We're also on YouTube, Facebook, and Twitch. Our website is TWIS, TWIS.org, twis.org. Are we ready for the science? To dive in? Okay, let's bring the science. Okay. I just have to get it out of the way, top of the show, talk about it before we go any further. The natural formation, the Darwin's Arch, or the Arch of Darwin in the Galapagos Islands has collapsed. And a group of scuba divers who were on a boat actually saw it collapse. It apparently collapsed due to natural erosion causes. And instead of now being still called Darwin's Arch, it's now potentially going to be called the Pillars of Evolution, which I kind of like. It's got a nice ring to it. It's got a nice ring. The pillars, it's now two rock towers that come out of the water instead of a nice arch formation. However, it just got me thinking a little bit about humans' effects on things. Dan Rather made a post about this arch falling on Twitter, and I replied, "Ha, finally something that humans didn't have anything to do with." But in thinking about it, I was like, well, actually, erosion has probably increased because of our effects on climate change and increases in storms and heating and wind. And so we probably helped it along just a little bit in the last 100 years. But I'm just going to, stop it right there. Other news. China landed a rover on the surface of Mars. It has a mission to look for fossilized microbial life. And that's what it's looking for. This makes China the second country in the world to land a rover on Mars.
So
Only
wait
the
a second.
second.
So it's looking for fossilized? So
Yes.
it's basically just got a camera.
It's got a camera, and and rocks. It's gonna be. It doesn't have rocks. It's gonna be looking for rocks. It's gonna be drilling. It's gonna be digging.
Okay, it has a drill.
Yeah.
It'll be digging, and it has a
It's
camera.
gonna be driving around, checking things out.
It's going to go find Curiosity, shake its hand.
Yeah. Yes. So new locations on Mars being explored.
Nice.
Yeah, by Chinese robots. Now, additionally, this is my last short entry story just to get it off because these are just stories I didn't need to talk about a lot, but I loved them so much. There are over 50 billion birds on the planet. There's at least 6 birds to every human being on the planet, according to a recent survey that took place over the last decade involving citizen science efforts to be able to report on bird sightings. They looked at something like 92% of all the recognized birds on Earth that they were cataloging and estimating, because of course we couldn't actually count every single one. There are estimates being made. These estimates put house sparrows in the billion bird club. There's over 1 billion house sparrows on the planet. So every time you go sit outside at a restaurant and you're like, oh, look at that cute little house sparrow. There's a reason you're seeing house sparrows. There's a lot of them. They're very successful, along with European starlings, ring-billed gulls, and the barn swallow. Very common birds worldwide, all in the billion bird club.
I would have assumed, perhaps erroneously, that chickens were the most populous bird.
That is maybe something they did not take into consideration in their count.
Yeah, they're probably counting wild birds.
Wild birds, yes. Yeah. Wild bird sightings as opposed to the factory farmed kind.
Yeah.
Yeah, but I wonder actually if we were to look into the number of farmed birds, how many would be in there? I bet,
I bet
definitely chickens.
Even amongst the farm birds, it would be chickens for sure.
An extremely quick Google. There are 25.9 billion chickens on the planet Earth.
Okay, there we go. So they're well in the billion bird club.
That's over 3 chickens per person.
Is that enough? I feel
we are
like
set.
It's not enough.
I mean, everyone can have mine, so I think you are okay.
You're set.
Still feel a little light.
Get your chicken.
Still feel a little light.
Chickens.
Even with all 3 of your chickens, still feel a little bit light.
Everybody gets at least 3. Come on, everyone gets their 3 chickens. I'm not going anywhere without my 3 chickens. Except
it needs to already
maybe,
be in the pot.
Yeah, but maybe I'd be finding other kinds of life if I lived on a different planet. Justin, you want to tell me about life on other planets?
Oh, that's sounds like an intro to a story about scientists searching for extraterrestrial life.
Yeah.
And we have talked about this from time to time on the show. Life extra elsewhere might not look like life on Earth. It might be different. It might have slightly different constituents. So if we have sensors that we've designed here on Earth with our expectations of what life should look like on Earth based on our one-off experience, we might be missing life elsewhere because our sensors aren't looking for how it could possibly be different. This is a new study by a joint Japan and U.S.-based team. This is led by researchers at Earth Life Science Institute at the Tokyo Institute of Technology, and they've developed a machine learning technique that can assess sort of complex organic mixtures using mass spectrometry and sort of reliably classify them into a biological or abiological category. And they took a bunch of basically what they did is they've fine-tuned this with a bunch of things that they like built in the lab versus naturally occurring organic compounds that are associated with life to some organic compounds that haven't been associated with life. And they've gotten it down to about 95% accuracy using this mass spec, which doesn't sound like sounds pretty good to me, 95%. But if you get that 5% false positive or missing 5% out there in space, that could be a problem. But they basically, right now, the only ways that we are currently looking for life elsewhere, we are listening to radio signals from advanced intelligent civilizations, which we should also tell them we are now on YouTube and Twitch, not just radio. They can look other places. Also,
find us all sorts of places.
All sorts of places. They also look for the differences in atmospheric composition around planets and stars, and now we're like you said, we're on Mars, digging in the dirt, looking to pull samples directly from the soil to see if there's organic compounds. Curiosity rover suggested that there was organic compounds on Mars. It's not exactly evidence of life, but it does sort of tell you that there is possibly the makings. One of the things that we have learned is that while we have life on this planet that started in a particular way, it wasn't the only way that it could have formed. There are slight differences that life could have taken at the outset. The fact that we have life that all seems to be from the same origination point, as again we've talked about on the show, is likely because once any kind of life got established, the niches fill up, and then anything else trying to come along later after all of these sort of biological niches are filled is immediately starting at the bottom of the food chain. Mm-hmm. And so trying to build up from the first step of life on a planet that already is filled niche, has all the niches filled with predators, exceedingly difficult. Which is why, perhaps, the only way that life has formed on planet Earth is the way that it happened to have formed. Not because that's the only way life can form anywhere in the universe, but because that's what happened first here. And once it starts, kind of hard to have a second branch or another type of life sharing the same planet starting.
Right. Okay. So if I'm understanding this correctly, then it's the reason that we don't have multiple different life trees of different forms is that basically one tree took off, started using all the resources, taking control of niches, competition got going, and there's just no room. Something else would have to come in and outcompete to be able to make it, and
it would have to outcompete. Sort of like if you were a newborn, and now you have to take on every MMA fighter right, who's in the league just to get a shot at being noticed. I mean, because you have to that every part of that step of that organic life's evolution has to beat out whatever is there.
Yeah,
and it's too much of a competition. When life starts first, you have a whole planet to expand into, to find whatever resources are there. You can differentiate and tackle it in all these sort of different directions. And every time you arrive at a new resource, you're the only one. So that appears to be sort of what has happened on this planet. So they've created this system.
This just an aside, it still just makes me think about, why have we not seen, or where is the evidence for other kinds of possible starts that never got the start?
Yeah. And it's because they just wouldn't make it. They wouldn't have room.
It wouldn't have made
so,
it. Okay.
So no, and the thing is, you don't have to go backwards. It could happen tomorrow. It could have been a month ago that a absolutely new form of organic life began on planet Earth somewhere. Possible.
And then it died.
And then it got eaten. Oh, then it was eaten. It's like, oh, I like your
fuel.
chemistry. I like your chemistry. Right. Let me use that to fuel my well-established chemistry.
But I mean, it's just goes to show that the way that organic chemistry works on our planet, the way that life-supporting chemistry works on our planet is the way that it got moving fastest first, and then runaway competition for resources. Push, pull, push, pull, push, pull all the way.
And basically what they've done here is they've trained the computer algorithm to detect within a mass spec signature of what compounds are available. They've trained it to identify complex, what they're calling prebiotic chemistry. So figuring out what those having already figured out what those other precursor chemistries might look like, what other combinations could have been possible, seeking that within the mass spec. So it's a little bit tiered. Very interesting. It might be able to categorize places to look for life a lot easier, a lot faster. This is something that could be very easily mounted onto a probe that's traveling through deep space and taking analysis of things and sending it back. It's not a high resolution, high power necessity to run mass spec. So it's a nice tool that we can add.
Well, there is a lot to be looked at on our own planet. 'Tis true, especially googly eyes. I like googly eyes. Do you like googly eyes, Blair?
Yes, I like googly eyes, but do you know who doesn't like googly eyes?
Ooh.
Burbs.
Oh, no.
Yeah.
Burbs.
A team of researchers affiliated with several institutions in the UK and one in Estonia have worked together to create a buoy that will repel seabirds. Why? Because they don't want stuff pooped on? No. It's because, actually something much sadder, seabirds are caught in gill nets all the time. So literally hundreds of thousands, some people think over 500,000 seabirds a year are drowned when they are caught in gill nets. Because that's their whole thing, is that they dive quickly into the water to snatch up fish. And a gill net is designed to be invisible in water. So, and gill nets catch fish. So then the seabird goes in and they're like, oh, a fish. Mm-hmm. Then they're stuck in a gill net and they can't breathe underwater. So that's a problem. Aside from just getting rid of gill nets, which would be great, researchers are trying to figure out how to save seabirds at places where gill nets are erected. So to start with, seabirds, like many other creatures, avoid eye contact with other creatures. And so the googly eyes they found were a really good deterrent. And so they put them on this small pole, attached it to a buoy, and then on the top were these big googly eyes. They were big enough so that birds, even with birds with poor vision, like geese, I didn't know geese had bad vision. I'm going to tell them next time I pass them. You can't even see me right. Geese, even geese can see them. And so they kind of scare away these birds by looking like they're looking right in their eyes. And the waves that make the buoy bob back and forth give them this looming effect. Plus, the little the centerpiece of this thing makes it turn with the wind. So then as it turns slowly, birds in every direction get looked at by these googly eyes. I think this looks a lot like Johnny Five. I don't know if
it does a bit. Yeah.
Mm-hmm. Yeah.
Yeah.
From
Johnny Five from Short Circuit. Yeah.
Yes. So anyway, they selected several sites near the gill nets. They counted how many birds approached and how many attempted to fish. And then they set up their googly-eyed buoy, and then they counted birds. And over the course of 62 days, they found the number of birds that tried to catch fish near the gill nets dropped by about a quarter, about 25%. Not great, but substantial. That's a lot if you're talking about hundreds of thousands of birds. And that was for up to a distance of 50 meters in every direction. And what's really
Pretty good
interesting is
distance.
they also found that the birds were less likely to fish near the buoys, even if the buoys had been removed for up to 3 weeks afterwards. So they were like, I don't know, that thing's going to come back. So that's
a
one
pretty
way.
good deterrent.
Yeah.
Keep them away.
Absolutely. Also, just don't put up gill nets, please. And if you're buying seafood, if you can buy sustainable seafood, usually that means it was not collected via gill net. So you can look into sustainable seafood also. Anyway.
Fewer gill nets mean that we need fewer googly eyes to deter birds. I'm going to move away from sight, the sight of googly eyes, onto the sound. Well, if you...
Of googly eyes?
No, not the sound. We'll get back to the sound of things later. But for people who are born congenitally deaf, there is no current way to give them their hearing back. If you're born deaf, your brain is going to develop without hearing. And for years, there was this idea that the proteins involved in the inner ear there's one specifically called otoferlin that is being studied by researchers at Oregon State University in biochemistry and biophysics. These researchers have been checking out otoferlin, and this protein was known to be involved in encoding hearing information. But the experiments that they just did using gene knockout technology, they were able to increase otoferlin's activity or get it out of the cells so that it wasn't there. And they could see how the hair cells, how the cells of the inner ear developed with and without otoferlin. They were able to determine that this protein that had only before been thought to be involved in information encoding, that sound transferring the vibrations into nerve signals, that is actually involved in sensory cell development. And without otoferlin, the sensory cells mature less quickly. They don't mature as well. And so now they're trying to figure out how they can use a smaller form of the gene, a truncated form of the gene, to be able to introduce it as a new mutation, like to using CRISPR, introduce it to people's ears, maybe to young children's ears before they've passed the plastic period of sensory development and hearing development, and potentially fix the otoferlin protein in people who are congenitally deaf. Now, they haven't done anything in humans yet, but in their study that was published in Molecular Biology of the Cell, they tested whether there's a portion of the protein that's necessary in being able to get this new mutation into the sensory cells. And so they did find that there's this particular transmembrane domain, and a transmembrane domain is a part of the gene that goes across the cell membrane. So there's part of it outside and there's part of it inside the cell. So it's definitely involved in some kind of cross-membrane ion channel transduction, some ion movement across the membrane. But anyway, they're studying it in zebrafish at this point. They're transparent and they don't have to be in big tanks. They just have to be in these small amounts of water. And they were able to find that a lack of the membrane domain led otoferlin to properly link in the way that it was supposed to link the neurotransmitter to the cell membrane. And so less neurotransmitter was released. And they say, our study suggests otoferlin's ability to tether the vesicles to the cell membrane is a key mechanistic step for neurotransmitter release during the encoding of sound. And so this whole study potentially is going to lead researchers on a better understanding of how hearing develops in the ear, in the brain, what proteins are involved, how they're involved, and potentially be able to treat mutations in otoferlin that lead to deafness at some point in the future.
Neat.
Yeah. So it's very interesting. I'm super excited and stuff like that. The idea that very isolated genetic modification could take place in the future, where maybe it's eardrops or maybe it's just a small procedure that where doctors go into the inner ear to apply the treatment, and suddenly you have a fixed cell. Suddenly you're able to turn vibrations into sound, into those nervous signals that your brain can then pick up and translate. Yeah, so very exciting. Down the road, nothing yet, but it's incredible information.
Very cool. I seem to have acquired the lighting director from the film Casablanca.
That's
If you
fantastic.
can see the lighting scheme of backdrops made of shadow, as there is this very bright light being reflected actually off an entirely...
It burns.
It's not even direct. It's bouncing off of a building right now. It's crazy.
That's a lot of fun.
This next story is kind of is also fun. What makes a great student? Would you say?
What makes a great student?
Yeah, what makes somebody a great student?
It depends on the teacher. Objectively,
you're
or
not in school anymore. You don't have to keep kissing up to teachers.
Okay, all right. I think asking questions is really good. I think that
and engaged. If the student is engaged, is paying attention, is curious, and wants to gather more information.
Okay.
Yes. I mainly mean because some teachers hate it when you ask too many questions.
Is that right?
I know from experience.
Okay. So, what's the answer?
Is it the agile mind, able to race ahead of the lesson and pull conclusions from the content as if the student were in conversation with the ancients from whom the lessons of knowledge were derived in the first place? Nah. Is it the spark of enthusiasm you're talking about? The student shows for the work, their ability to work well with others in the class, and the mark of being prepared to be a good future employee once they graduate? Nope. That's not it. Is it the mind for being academically efficient, getting good grades, and knowing how to operate within a read and regurgitate system of education? No, that's not it either, according to, at least not in the eyes of typical university staff, according to the Journal of Educational Review. The ideal student is punctual, organized, and works hard. That's it. That's what they want. They want you to come in and do the schoolwork and be on time and have your stuff together when you get there. This is the result of a new study that involved a survey of 1,000 students and staff at British universities, as well as a focus group with 132 members of both of those groups. Participants were asked to write down the top 5 and least most important characteristics of an ideal student. Interestingly, that list was slightly different between the two groups. The academic skills, employability skills, and intelligence and strategic approach were ranked in the bottom 3, actually by the staff and the students. Although the students did rate employability skills much higher than the teachers, presumably because they may want to get jobs as a result of their education.
I want to know what the student body size is of these universities and what their class sizes are. Because if you are in a class of 20 students, the student who is asking questions, who is engaged, who has an interesting conversation around the topic you're discussing is going to pop out and way more than the person who's organized. But if you are assessing your students based on 200 essays you've collected from your lecture hall, and you get almost no interaction one-on-one with those students throughout a semester, your rubric is very different on
how
which
easy is
it
to make a comment based on how organized they were in formatting their paper or whatever.
I want you to listen to some sounds and tell me what words you think they represent. Okay, ready for this?
Okay.
Okay.
Wait, what? Can I get it one more time?
Wait, are we like? Is this like charades, but with
It's
sound?
Like with sounds. It is sound
This
charades.
is one of those things that until we're like, we won't be able to know what it is until we're primed. And then we'll be like, oh, we know what it is. And then they go, oh, well, actually, it's this, but you were primed the wrong way. It's like one of those psychological.
Sounds like a can opening. Okay,
so you've got a can opening. All right. Any guess, Justin?
Checkers? I don't know.
Checkers. All right. We're going to move on to the next sound.
That's definitely a can opening.
Yeah.
No. What is that? That's somebody saying "rawr."
Yeah. So I could go really literal and say like tiger, or I could say fierce.
Okay. Ooh. Good. Okay, we'll go to the next sound.
I hate that. So my word is hate.
Oh, are we just supposed to associate a word with it?
Yeah, what does this word? It's word charades. This sound. What word is this?
No, stop it. Don't do it again.
Wait, 3 more times? Just because I didn't hear it that time, because somebody was screaming over it. Blooper.
Blooper. Okay, we got a blooper from Justin and hate from Blair. And the final sound.
Ha'ing.
Hooray.
It's ha'ing.
So Blair says hooray. What is... Okay.
Ha'ing.
I will not keep anyone waiting any longer for the answers here. The first sound is meant to represent the word cut. So can opener was very close.
All right.
Cut.
I see what's it. I needed an example. I'm off.
Yeah, Blair.
Very well done.
You totally nailed tiger.
Oh, good.
Nailed it.
Tiger.
Nailed tiger. That was that sound. The second sound was tiger. The third sound that Blair hated was water.
That's not water. That's like gloop.
Yeah.
Supposed to be water.
It's like bubbling mud.
And the last sound was good. It was supposed to represent the word good. The reason I'm bringing all of these sounds is that researchers from the University of Birmingham and the Leibniz Center General Linguistics in Berlin discovered that iconic vocalizations, so sounds, not words per se, but sounds similar to body movements that have that represent words and have gone on to create sign languages and even languages themselves, that there are iconic vocalizations that convey a wide range of meanings. And they used sounds like this for these vocalizations for 30 different words, different meanings that are common across different languages. And they looked into people across 28 languages, 12 language families, groups from oral cultures, like speakers from the Palikir living in the Amazon forest, speakers of the Daki in the South Pacific island of Vanuatu. There were a number of people, and they say that listeners from each language were more accurate than chance at guessing the intended referent of the vocalizations for each of the meanings tested. Now, in the test, it was a bit easier than what I just played, just playing the songs, well, not songs, but the sounds at you. They were played the sounds, and then they could choose a word from a list of 5. So they had a 1 in 20, a 1 in 5 chance of getting it correct. And so, but they picked across languages, universally, these vocalizations seem to underpin meanings. And so the researchers suggest that their findings challenge the idea that vocalizations have limited potential for iconic representation, demonstrating that in the absence of words, people can use vocalizations to communicate a variety of meanings. I mean, we've got
Okay.
sounds like, huh? Meh.
Yeah.
Yay!
Yeah.
But
Aw.
Aw.
but I think it's a very specific room full of nerds. We're gonna get.
I feel like you could say that
It's
about
happy.
literally
Uh-huh.
any story on this show.
That happy is not that one I didn't buy for sure.
That guy was happy for sure.
I'm confused.
He wasn't happy. It was good. It was like
Good. It was good.
Okay, it was just good.
good.
Yeah.
Right.
Woo.
Anyway,
There you
it's
go.
a lot of fun.
Exactly,
Potential
Rosa.
for
Woo.
vocalizations, sounds
Iconic
to
sounds.
have predated and maybe been ancestral, probably in combination with body movements to represent ideas. There were probably vocalizations and movements, physical and auditory charades that were played by early people to create languages, to create communication that was shared across individuals. So it's just a lot of fun to be able to play this kind of game.
Well, and think about communicating with somebody who doesn't speak your language. They're talking at you in their language, or you're talking at them in your language, and you can often get meaning across with the combination of inflection and body movement. And inflection is at the basis of what this is about. It's about sounds that convey meaning, even if you don't understand, if it's gibberish to somebody.
Yeah. This is TWIS. If you just tuned in, thank you for listening. If you've been listening, thank you for listening. I do hope that you're enjoying the show. And if you are enjoying it, please share it with a friend. All right, everyone, time for the quick COVID update for the week. Boop, ba-da-loop, boop, boo. This week, the CDC is kind of going through it after they completely switched directions on their mask guidance. Not 1 week prior, the head of the CDC had been in front of Congress saying, we got to wear masks. Masks are important. It's really good. And a week later, the CDC changed its guidance to say that vaccinated individuals can go without masks indoors and outdoors, and except in places like hospitals, healthcare locations, or on public transit, public transportation.
Or any private place that's
place.
still saying to put your mask on.
Yeah,
You
but
still have
what
to
it is
listen.
the hornet's nest is basically that it punted the whole situation onto business owners to decide whether or not to check for vaccination, whether or not people have been vaccinated upon entry. But now we're getting to a point where people are required in some locations to wear masks to enter indoors locations. It's great. Wear your mask. If you're vaccinated, wear your mask. Or if you have your card and you can show that you're vaccinated, if you can prove it, do that. If you feel comfortable not wearing a mask and you're happy doing that. But I mean, don't make a big deal out of it. Let's just try and make it easy on everybody. I
think the real issue is that 30-something% of the United States is vaccinated. So
we're at 37% of the United States that are vaccinated. And that I do think you're right, Blair. I think that was one of the other issues, is that just the week prior, she had been saying that that we would continue to wear masks until vaccination rates were widespread. Everybody, we're starting to head toward higher vaccination rates, lower case rates. And it seems as though they've just given up. They had said that they were going to wait until we were below 1,000 cases a day across the country. And they've just completely given up on that.
Also knowing that we're about to hit the summer craziness again. And if we look at, I realize people are vaccinated out there, which is great. But if we look at anything like infection trends from last summer, we are about to go into a period that could really be problematic. So I'm not I'm not thrilled. The timing seems less than perfect.
Yeah, and from a science brief from the CDC themselves, they reference a study that says, in one study, complete relaxation of prevention measures prior to adequate vaccination coverage resulted in essentially no reductions in SARS-CoV-2 infections. And this was a study in which they simulated various vaccination rates and different non-pharmaceutical interventions, mask wearing, social distancing, all that kind of stuff. And they adjusted the percentage of these different things. And really, in their simulations, not real world, just a simulated study, if vaccination was not adequate and you relax the prevention measures, the SARS-CoV-2 infections stay high. They don't get reduced. It's a combination of things we have to deal with.
Absolutely. Right.
So, yeah. So, to be safe,
So,
when you're around other vaccinated individuals, you can be safe without a mask. But if you're around groups, crowds where you don't know the status of everybody else, you don't know if you have immunocompromised individuals, kids under the age of 12 who are not yet vaccinated, all sorts of people who are vaccinated, but for whatever reason, their immune system is not taking the vaccine as well, people can still be at risk. So let's all just calm it down and be nice to each other, wear masks in public.
This is why what they just did was exceedingly dangerous, because what was said was that if you're completely vaccinated and don't have COVID, you can be with somebody else who's completely vaccinated and doesn't have COVID. But nothing else was said. And the way it's heard is masks off, business as usual.
Everybody.
Send the workers back to the front line dealing with the indiscriminate public, because we're done having. So, hey, let's all have another wave. Let's have another spike. Let's fill those hospitals with more sick people, and let's have a lot more death, because gosh darn it, we have to open the kiosks.
And don't forget, in this, the communication, the science communication around this was poor. Absolutely. Yeah. And we have to remember that there are new variants that are more transmissible. We talked last week about how they're more transmissible in kids and who are not vaccinated yet.
No, they're not even allowed to be vaccinated.
Yeah, let's wear our masks. Let's do it for the kids. Let's do it for the kids. Another study. So we do know that the CDC is frustrating everyone. Guidance is not great. And even Fauci came out this weekend and said, be prepared for us to update our guidance, because they will probably spend a week figuring out why everybody's frustrated and then make up some new rules. Because, I mean, you hope that our governmental public health institutions have things in control, but everybody is still playing the new pandemic game, the we've never done this before pandemic game, and there are a lot of mistakes, and it's really unfortunate, because lives are at stake. Yeah. Another study, in contrast to a study that I reported on last week that posited SARS-CoV-2 viral integration into human DNA, there's a new study out this week in the Journal of Virology suggesting that SARS-CoV-2 cannot integrate itself into human DNA. So now we have one study which has gone through bigger reviews, revisions, that says, yes, SARS-CoV-2 is a retrovirus getting into your DNA. And then another study saying, nope, that is just an artifact from the way that the study was run, and the question still remains. So I put it to the virologists and the molecular chemists and molecular biologists to please figure this one out, because it's an interesting question. Is SARS capable? Are coronaviruses capable of hijacking cellular machinery to integrate bits of themselves into our DNA. Is it possible? Big question. And Justin, you have an interesting question as well that you wanted to talk about?
Oh, yeah. So there's 18 scientists from research institutions that are now urging a deeper dive into the origins of the coronavirus. They published last week in the journal Science, and they're arguing that there's not yet enough evidence to rule out the possibility that SARS-CoV-2 virus has escaped from a lab. Why is this important? Because there's a report from the World Health Organization that came out that said it definitely couldn't possibly, no way that it came from a lab in China. Problem is, it was pretty much based on data that was being offered by essentially the Chinese government. So you have to take with a grain of salt any information that's coming from China about this, of course, because all information that comes from China has to be approved by the Chinese state. And these researchers have come out from various institutions and said, hold up, wait. This is David Railman, Dr. David Railman, professor of microbiology and immunology at Stanford University, who's one of the signers of the paper. We believe this question deserves fair and thorough science-based investigation, and that any subsequent judgment should be made on the data available. Also stating that they and their colleagues have looked at this, and even though that they're like experts in their fields, they don't believe there was enough information or data in this report from the World Health Organization that could actually come to the determinations that the report came to. Within that report, they found that the chance that it was an accidental laboratory leak was deemed extremely unlikely. The likelihood that the virus jumped from a source animal to an intermediary species and then to humans was considered likely to very likely. Other pathways that were investigated, direct jump from animal to human without the intermediate host was considered possible to likely. Transmission from the surface of frozen food products was possible. Oh gosh. Ravinda Gupta, a professor of clinical microbiology, University of Cambridge, who's also a signer of the letter, said he would like to review lab notes from scientists working at Wuhan Institute of Virology. This is a research center where the coronaviruses are studied. It's in Wuhan, which is why there's a pretty decent interest in that lab as being a potential source of its exposure to humans. They'd also like to see the list of the viruses that have been used in the Institute over a 5-year period, something that has apparently not been made transparent, which is a thing that you would think would be curious. A point of interest at this point. Tedros Adhanom Ghebreyesus, Director General of the World Health Organization, actually expressed a similar opinion when the report was released, saying, although the team has concluded that a laboratory leak is the least likely hypothesis, this requires further investigation, potentially with additional missions involving specialist experts, which I am ready to deploy. Let me say clearly that, as far as the World Health Organization is concerned, all hypotheses remain on the table. So even though the World Health Organization sort of put out this report, they still haven't completely signed off on it being the end-all, be-all of the situation. Another thing to keep in mind, too, is while this is an ongoing thing and we don't know, one of the things that we have not found is any animal source. This is
right.
There were something like, I got it in here, something 80,000 wildlife, livestock, and poultry samples have been collected from 31 provinces in China. None of them turned up a SARS-CoV-2 antibody or snippet of the virus's genetic material before or after the outbreak. So thus far, after what sounds like a pretty extensive search so far, could have been missed. It's still out there. We could easily have missed it. But having looked at that and not found it, it makes you think there might be another source. If it was collected in a lab, if it's in this lab, it could have exposure to humans over a prolonged period of time of working with virus that might otherwise not have been something that was infecting humans right away. It could have been something that was an exposure over time. And any lab keeping a virus in it is very difficult. They're tricky. They can sneak a ride on humans if the
they can sneak a
PPE
ride,
isn't perfect.
And part of coronavirus studies is that they're usually not BSL level 4. They're not biosafety level 4. They're which is the highest level of sanitary, keep it in a bubble kind of protections. Usually they're level 2 or 3. So there is a little bit more. There's a bit more relaxation on how techniques are done, the safety precautions that are in play. And they have had viruses escape or go out previously. There have been American labs that have had viruses go out with people from BSL level 2 and 3 labs in the past. So it's not to say that it hasn't happened ever, that things have left the lab. And I've also read that in the scientist notes that have been provided, the communications that have happened, there isn't evidence of this exact strain of a SARS coronavirus being in the laboratory. Like, that's not something that they were working on. So it's a big question. Where is it if it was in the natural population? If we want to find it out in the wild, where's that reservoir? Where are we going to find it? What animal species did it come from? We have no idea. If it was from a lab, there are outstanding questions. At this point, I think I've seen more evidence to the contrary, but the report found it to be like the least likely option. It's still, it's science. So it's like, this is the least likely scenario, but we don't have all the evidence to concretely say it didn't come from the lab. There's still, there are some possibilities, but most of the evidence suggests it did not. So, and anyway, yeah.
I don't know that that's true at all.
But we should researchers should continue to investigate the question of the source, because it will help us understand this whole pandemic and SARS coronaviruses even further. And that on its own is important if it can protect people moving forward. That does it for our COVID update. This is This Week in Science. If you are enjoying the show, please consider supporting the show. You can find our Patreon link at TWIS.org. Click on that link and choose your level of support over in our Patreon community. $10 a month and up. And we will thank you by name at the end of the show. There are also other fun rewards for being patrons. We can't do this show without your help. So thank you so much for all of your support. It's time now for the wonderful animal breathing, air-filled portion of the show. Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
Blair.
How much do you like to breathe?
Oh, all the way, all the time.
Everyone can take
respiration.
A deep breath.
I love it. I don't like it when my sinuses are full.
Take a deep breath in. And out. Doesn't that feel nice?
Yes.
What if you could breathe another way? So this is
suddenly an episode of South Park.
Yes. So this is a piece of research looking at the delivery of oxygen gas or oxygenated liquid through the rectum. This is a look at artificial respiration, artificial respiratory support. We already use it in other ways, like you get intubated and stuff like that, as clinical management of respiratory failure, which could be as a result of pneumonia, acute respiratory distress, any of those things related to COVID, for example. And so the ability to go in and medically assist with respiration is very important. So Takanori Takebe of Tokyo Medical and Dental University and the Cincinnati Children's Hospital Medical Center wanted to look at using the rectum for artificial respiratory support. The inspiration for this come from sea cucumbers, freshwater fish called loaches, certain freshwater catfish. All these animals use unusual forms of respiration, many of them their intestines. It's a very absorbent surface. And so the idea here was to provide evidence for intestinal breathing in a mammal so that we could try to kind of translate that into human medicine. So this is looking go ahead.
I just need to jump in here
Yes.
and comment on the use of the word breathing.
Yes.
Because when you think
Fair
of
point.
breathing,
Yes.
it is
Yes.
the air,
Yes.
your lungs inflating and deflating. Breathing is your respiratory system process. It's not specifically just respiration. Respiration is a metabolic process, which is the transfer of oxygen and carbon dioxide. Your cells respire, right?
Very good point. I used breathing for comical effect, but I think it is a very
Everybody's
important. Yes.
Everybody's using breathing in
Yep.
the headlines because it's like what people understand,
Yes.
and I get that.
The lungs
but-
are not involved. There are no alveoli used in these processes.
And this is not allowing animals or people to breathe out their butts.
Right. Unlike turtles we've talked about on this show before.
Yes,
they can breathe through their butts
can.
because it is a passive thing that is just part of their normal processes. And so
Yeah.
that is what's happening in the sea cucumbers and these fish is they are breathing through their intestines because they're in water. They live in this stuff and they are saturated by it. And so they can pull oxygen out of the water. So even then it's not breathing, it's respiration. But that's not even what this is about. This is a step even removed from that. That's rescue respiratory assistance. So there's a difference from that. So we have breathing, which is using the lungs and you're using that to pull oxygen out of air and push it straight into your, okay, so that's all happening. Then there's respiration, which can be done by pulling oxygen out of water through something that is not a gill or a lung. In this case, you're just using membranes through the intestine. And then there's this third thing that's even a step removed that is a kind of manual, how do I put this without it sounding really upsetting? There's no way. Forced contact with oxygen in the rectum to aid someone who does not have good oxygen levels. So here's the actual study. They designed an intestinal gas ventilation system to administer pure oxygen into the rectum of mice. So they're just putting air up there. Without the system, no mice survived 11 minutes in extremely low oxygen conditions. They all fell short of 11 minutes. But when they were ventilated rectally, 75% of mice survived 50 minutes in normally lethal low-level oxygen conditions. So that's huge. So that's step one. So that's just air, right?
Mm-hmm.
But this is not going to fly for human medical care because intestinal gas ventilation requires abrasion of intestinal mucosa. If you're not a doctor, you can tell that sounds bad. You don't want that. So it's not clinically feasible. It's not good to push a bunch of air up there. It's not what it wants. It's not good for the bacteria that lives there. It's not
Abrasion. You're just disturbing the
membrane.
mucosal lining. Yeah, it's going to mess everything up.
It's bad.
Bad.
So Instead, the researchers wanted to look at a liquid-based alternative. So they used oxygenated perfluorochemicals. They've already been used in other medical processes in humans. So they're already biocompatible. They're already clinically approved. So they used intestinal liquid ventilation in rodents and pigs via this liquid perfluorochemical. They received, when they received this intestinal ventilation, they could walk farther in a 10% oxygen chamber, and more oxygen reached their heart. So also their skin looked better. They were less cold. Their skin had more oxygen in it. They didn't see any side effects. So this all looks good. So the next plan is to expand their preclinical studies to pursue regulatory steps to kind of figure out what potential side effects there could be and how they can do this in the best way possible in order to move to clinical translation. So the lead researcher, Takebe, says, the level of arterial oxygenation provided by our ventilation system, if scaled for human application, is likely sufficient to treat patients with severe respiratory failure, potentially providing life-saving oxygenation. Mm-hmm. So if, for example, if you have pneumonia, if you have pneumonia, or if you have a collapsed lung, if your lungs are not working for whatever reason, intubation is not really going to help you. So how can you get oxygen to a person whose lungs are not currently working? This is how. And so this study might seem very silly, talking about breathing through your rear and all this kind of stuff, but really, this could be life and death for people in hospital situations. So from that, I want to talk about scuba diving lizards.
Of course, because we're going to go. Okay, yes.
Yes. I mean, they're connected. It's all about oxygen. So this is a team of evolutionary biologists from the University of Toronto looking at anoles type of lizard. They are able to breathe underwater. And it's been observed that these guys can dive down underwater for a really long period of time. And there's been a lot of kind of observational stuff related to that. And we know, for example, that they can dive underwater to avoid predators, and they can remain submerged for as long as 18 minutes, which in the grand scheme of the animal kingdom is not crazy long. But if you think about just a normal terrestrial animal, that's a very long time. So how are they doing that? It looks like they're actually, they make their own scuba apparatus. And by that, I mean, they use their hydrophobic skin to breathe out a little bit out of their nose, and they exhale a little bubble that clings to their skin. They then re-inhale the air and back out and back in and back out and back in. We know the oxygen doesn't all go away the first time you breathe in a breath of air. So there's a reserve of oxygen that they're working off of in that bubble. So they're calling it a scuba situation because it's like a rebreather on a scuba tank, right? And so the way that they checked on this is they're not just blowing bubbles for fun. They measured the oxygen content of the air bubbles, and they found that it did, in fact, decrease over time. So they were rebreathing the air back in and out from this bubble over and over. And of course, this helps them avoid predators longer, right? So this would be an evolutionarily advantageous thing to do. And this all started with just some observations back in 2009 and then later in 2019, but they really wanted to test this. And in the end, they still have some interesting questions. So they have some funny hypotheses for future study, which is really what I thought was interesting about this. Is for one, they think the reason they're so good at this is not just their hydrophobic skin, but also that they think they push out the air from their nasal passages and mouth and windpipe, which don't make contact with their lungs. So they're able to basically use this air that's not getting oxygen pulled from it anyway to be able to push it out with what's in the lungs and mix it all up and pull it back in. So it's less depleted because they have that reserve in the rest of their body. But they also think that it helps them rid their body of carbon dioxide. They have a hypothesis that the carbon dioxide in the bubble from their lungs dissipates into the water, because there have been previous studies that CO2, because it's so soluble in water, and because the level of CO2 in the bubbles is higher than the water, it follows the gradient. And so it actually leaves the bubble and goes into the water. And in turn, they think that it might even act as a gill to absorb water from or oxygen from the water, which is something that aquatic beetles do, by the way. They use little bubbles, they exhale, they have this gas exchange, and then they pull it back in. So they really want to look into this and see how intense this bubble is helping them as a scuba tank.
I find it amazing. I mean, it's one thing for an invertebrate beetle to use a bubble like this for respiration, but for a larger organism, these animals, they're small, but at the same time, this is this has to be a significant bubble and process for them to be able to use it like a rebreather, to be able to swim underwater for such an extended period of time. Like that, this is a big deal. How come I don't know how to breathe a bubble out of my nose that I can rebreathe with?
So admittedly, while I was first reading this story, I kind of like breathed air out into my mouth and then breathed it back in and out and in and out and in. And I don't think it helped me at all.
No.
So
And
I must be missing something.
Anyone who has tried to inhale the air out of a balloon and blow it back into a balloon and use a balloon as a rebreather, you find that you pass out fairly quickly. That you very quickly deplete the oxygen in that balloon and fill it with carbon dioxide.
Why is that though? Why are we so much, I guess, better at breathing? I guess because we're so big and metabolically complicated. But yeah, it's pretty wild. And of course, they had to throw in here, the researchers mentioned that it's too early to tell if lizard rebreathing will lead to any particular human innovations, but biomimicry of rebreathing may be an interesting proposition for several fields, including scuba diving rebreathing technology, which motivated our naming of this phenomenon. So, yeah, it's possible, but also I think, isn't that kind of what they already do?
Yeah. Yeah.
I guess the part that we would want to key into is the pulling oxygen from water and releasing carbon dioxide. But I don't know. Yeah,
that there is a diffusion of oxygen from the water into the bubble, into the body of the animal, right? And the diffusion also of the transfer of carbon dioxide out into the water. And so how do you turn a bubble into a membrane that can make this work? I don't get that. How do you create the diffusion gradient that allows that to happen, but not it's extending your lung yet with just this buccal cavity with this? Yeah, I think that's the thing I'm most interested in regarding this study, even though that's the thing they haven't actually studied yet. But stay tuned. Stay tuned.
Maybe around episode 1000, you'll hear all about it.
Stay tuned for more scuba diving lizards on future episodes of This Week in Science. All right. It's time for Justin to tell us some science.
Oh, what do you want to know about? Let's see.
You're going to extend the animal corner with some more animal stories.
Yeah, right? Let's talk horses.
A horse is a horse, of course.
Yeah, a new study of ancient DNA from horse fossils found in North America and Eurasia shows that horse populations on the two continents remained connected through the Bering Land Bridge, moving back and forth and interbreeding multiple times over hundreds of thousands of years. So The new finding was based on genetic continuity between the horses that died out in North America, which was not very long ago. That's about 11,000 years ago. That's before Egypt. That's before current population of Europe was in Europe. This is time when the Americas were actually still, that Native Americans were populating pretty much both North and South America. 11,000 years ago, North American horse died out and then gets reintroduced later by Europeans again. So this study is in molecular ecology. It's available online. Corresponding author Ben Shapiro, professor of ecology and evolutionary biology at UC Santa Cruz, says the results of this paper show the DNA flowed readily between Asia and North America during the ice ages, maintaining physical and evolutionary connectivity between horse populations across the Northern Hemisphere. So this Bering Land Bridge that we sort of have thought about, I always sort of picture it as a series of little islands between Alaska and I guess Siberia in Russia. But it's actually it's much more expansive than that. From it gives a couple of landmarks here, from the Lena River in Russia to the Mackenzie River in Canada. There were extensive grasslands between those two places because so much of the water that would have been in the ocean was now in ice on land somewhere with these massive glaciers that had formed. And so there was this place where horses and mammoths and bison and other Pleistocene fauna were just moseying about. And in this area in between, these horses that had gotten separated about 1 million years ago were able to get back together, to breed again, to switch sides, maybe even. Anyway, this result is showing that the North American horse that had gone extinct is maybe a little bit more of a reintroduction to North America than it is an invasive species, perhaps. Anyway, new findings are going to fuel controversy over the management of wild horses in the United States, as descendants of domestic horses are brought over by Europeans. Many people regard wild horses in the United States as an invasive species. Others consider them to be part of the native fauna of North America at this point. But whatever the white people in America decide about what constitutes invasive or not will likely be taken with a grain of salt by people who are actually native to the Americas. And in other news, memory is a weird thing. We all have memory. Yes?
Huh?
Huh? We all have memory?
I don't remember. Do I?
We all have memory of some form.
Have I learned anything in my life? Do you have memory?
We all have some form of ability to recall things, but we don't all have the same way of doing it. And we don't really talk about it a whole heck of a lot, about how our memories operate. Have you ever met somebody who has a photographic memory? Either of you?
Yes and no. I have a friend who has a photographic memory for dates. She's able to remember exact dates when things happened.
Okay.
Which I've always found fascinating. How do you do that?
I've met one person who actually has like a freakish photographic memory where they can look at something and recall it days, weeks, a month later, no problem. And the way it works with them is it's like they're taking a snapshot, like a picture, like photographic memory is the exact right way to define this. They're taking a picture of it, and it's not that they're recalling what it said. It's that they can be reading it for the first time a month later because the text is saved, right? The visual is saved in their head, and they might not know the information, but they can read it from the memory.
So this is all long-term versus short-term memory storage? Is that what you're talking about?
It's just memory. It's just however you want to regurgitate something that you have learned. So anyway, compared to that sort of memory, though, that ability to take a picture of something and recall it at any point later. Most of us need a lot more help in remembering.
We need techniques.
Yeah, we need some techniques. So some Australian scientists looked into an ancient Greek technique of memorizing, which I actually heard about for the first time in a Sherlock episode, which is the Mind Palace.
Mm-hmm.
Oh, the Mind Palace. And that memorization technique is basically one of taking like a childhood home and placing bits of information around in the mental memory blueprint of that house so that you might associate certain information with certain rooms or areas of that house. Something that's already well imprinted in your mind, the rooms of the house or where the bathroom is or something like this. You can lay other information on top of that and apparently get a pretty good recall out of it. So they were studying that and they compared it to a memory technique developed by Australian Aboriginal people, which have a very interesting, because they have a word of mouth tradition, an oral history. So they have to pass everything down over 50,000 years through that oral history. So memory becomes very, very important. Got to remember the stories of the ancients in order to retell them so the next generation can hear them, that sort of thing. And what they do is actually very similar to that mind memory palace technique, where they have areas of the terrain, areas of the land associated with the information. But then they also take an extra step and they build a little story around it to go with it. So they tested these two techniques, as well as they had a third group of these were all medical students who they were testing these memory techniques on. And they had a third group that basically like watched a video on thumb twiddling. And then they gave them a bunch of information on butterflies. They tried to find something that wasn't, something they were already studying, but they threw a whole bunch of butterfly names at them. And they came back and tested their recall at 10-minute and 30-minute intervals based on techniques. Turns out the Aboriginal technique with the narrative and the sort of locations was the most effective, as folks were 3 times more likely to correctly remember the entire list than they were prior to training on the technique. The students using the memory palace technique were twice as likely to remember the list perfectly if they had than they had before they had studied the memory palace technique. Interestingly, there was about a 50% improvement over the pre-training performance by the group that watched the thumb twiddling video or whatever. So thumb twiddling ain't for nothing, is what I'm trying to say. There's something to that as well. But more importantly, the survey found the students who used the Aboriginal technique also found it the most enjoyable technique.
Yeah, I mean, the memory palace technique, I've always thought that in the memory palace, you're supposed to, you put things in places in your memory palace. So say you choose your house to be the palace, and you go around your house and you put things in a particular order in the house, and you're supposed to also associate them potentially with something kind of bizarre. So that's part of the memory palace technique, where that kind of bizarre association, because of the novelty of it, also allows the item to be associated with the thing that is already in your memory, this very deep memory of a place that you know very, very well. And so that kind of that novelty, that fits really well with this, what you're saying about the Aboriginal memory technique of telling a story. Because along with the story, you're creating context and you're creating novel, unique aspects, details that are going to link all of these items into the locations in the memory palace, in the landscape that are there. So it all, I can see how that added detail, the contextualization, the giving the brain more to chew on as a reason to put things down in memory can help you go through it. All right, let's dive into some brainy news. Move past the memory palace.
Kiki,
Yes?
you brought a brain story?
I brought a brain story.
What?
That's surprising, isn't it?
Oh my god, it's Kiki's brain corner.
I thought we were taking the week off. I got to find a Neanderthal story.
I brought one brain story. The thumb brain. Your brain will expand to accept a new thumb. Apparently, researchers looking at a new prosthetic thumb, a robotic thumb that's being developed by researchers at the University College London. They published in Science Robotics about their study to see how this robotic third thumb, how it affects the brain. Because a big question is, how do our brains take in new devices as we learn to use them, and how does our brain change to accept these kinds of devices? So the research started with the design of this quote unquote third thumb by a designer, Danny Claude. She designed the third thumb to latch onto your wrist and hand, and to, for all intents and purposes, stick out of the other side of your wrist as if it is an additional digit that can be used. And it is controlled by controls on the feet, that you can move your feet around to control the movement of the thumb. And it's this really interesting device. They trained a bunch of people on how to use this third thumb to do all sorts of daily tasks, manipulate objects. And in the process, they said, hey, let's put these people in an fMRI and see what happens to the motor cortex of their brain. Usually, before the use of the third thumb, each of the fingers is very distinct in the motor cortex of the brain. It's like there is a spot for your thumb, your first, second, third, fourth, and fifth fingers. So there is a specific spot where the neurons are active to move and use each of your digits in your brain. Upon the integration of the third thumb, this distinction went away, and it became more of a finger area in the motor cortex.
Interesting.
So the finger area de-differentiated to a certain extent.
Wow.
Yeah. And it seemed as though even after training had taken place and individuals had stopped using the thumb for a week, that de-differentiation, it stayed in the brain. So the brain
Were they still able to type?
Yeah, they're still able to. Yeah, all the normal stuff is still happening. But it's fascinating that the brain was plastic enough to pick up the new digit. You know, and we pick up new devices all the time. But pencils, we drive cars as extensions of our bodies. We are learning
vacuum cleaner, even.
Vacuum cleaner.
Anything.
Yeah. Anything is an extension of our bodies. We learn how to use them, and we learn how to manipulate them. But in the case of something like a prosthetic, to either replace missing limbs, or in this case, add limbs, add a digit to the body, the question is, if you're adding on, how does the brain deal with that? What if you wanted to have multiple robotic arms that you control with your brain, or that, how does this get integrated? How much can our brains handle? And what's interesting to see is that the brain can handle the extra thumb. It learns how to use it, it integrates it, but there's that plasticity, but at the same time, it affects the way that the brain interacts with all of the fingers. But this was a short period of time that
right.
the training...
So maybe given more time, it could have
maybe it would have differentiated back, and maybe it would have added a new digit, a new thumb into the motor cortex. We don't know that.
So you know what it makes me think of is like old school video game controllers, like a Sega Genesis that had like 3 buttons and a directional pad, right? Mm-hmm. And if you were someone who used that and only that, and then you handed them an Xbox controller with like triggers on it and all this different stuff, right? Then I bet you would see similar stuff happening in the brain where you'd see completely new areas lighting up from when they were using that initial controller. And then eventually it would kind of settle back into the normal video game space, right? Because, I mean, I think adding on extra stuff, at that point, it's just a tool, I feel like.
But it's interesting because it is a tool, but so in the case of a video game controller, it's all the motions that your thumbs are normally going to make. It's just learning how to coordinate them with the video game, right? So a different controller, you're just learning that coordination for a different game, different controller. So that's going to be a learning process of just motor coordination. But in the case of like a full additional thumb that you can use in addition to your other thumbs, I mean, that's more than just coordinating in a certain extent.
I feel like if this connected to your brain, it would be a better test.
Right. It's not directly connected to the brain in this. Yes, absolutely.
Because I think that's the part that I keep getting stuck on, is because it's kind of a pedal-based thing.
Yes.
It still feels kind of just like a tool.
Right. I think you're right. Yeah, absolutely. Yep.
So that's good. That makes me think, if I was operating this somehow just with my big toe.
Shouldn't the space in your brain for your big toe become bigger? No.
Yeah, I know. Like, I feel like the big toe, like the fingers could have stayed differentiated. And the big toe's like, I got this. Let me just throw in. And it would be like, anyway. I wonder if my big toe is differentiated from the rest of my toes. I don't feel like the rest of my toes have their own motor.
Your big toe probably is, but the other ones,
the other toes are
just
yeah.
It's when the doctor tells you to wiggle your toes, and you're like, "How do I even?
How do I use these?" Yeah. Yes, yeah. So it's an interesting study. I think there's a lot more work that could be done there, because I mean, the future of augmenting the human form we're just getting started with that, and it has the potential to be very interesting, depending on how many of these tools we can integrate. How much can our brain do? How plastic can it be? And what happens?
Like an extra thumb does, though, sound like something that like is like a wish of a Neanderthal. It's like an old wish. I don't know that that's as crucial now. Oh, maybe it's becoming
I will tell you, yes, as someone who has nerve damage in my right hand and drops things all the time, I would love a second thumb on my right hand. Great. It would be so helpful.
Would you put it on the same side?
No.
Just to replace the thumb? I was just checking.
No, what am I, a koala?
Do they have 2 thumbs on one side? Is that a koala thing, really?
Yeah.
Oh, I had no idea.
Yeah.
Oh.
Koalas, they're all thumbs.
They are.
All right, my last story for the night is an interesting story related to the biochemistry of Alzheimer's and how Alzheimer's and the proteins involved potentially get their start in the neurons in the brain. Researchers at Massachusetts General Hospital were trying to figure out how amyloid beta gets produced. Amyloid beta is one of the proteins that, when it's all gunked up in the neurons, is thought to be a neurotoxin that causes Alzheimer's disease, or at least in part, a part of the cause of Alzheimer's disease. They've published in Cell Reports their study looking at the mechanisms of how this all works, and they dug into a bunch of questions related to where in the cell and how does amyloid beta get put together? All right. So first off, a long time ago, this team, the researchers involved, they discovered the first Alzheimer's disease gene called APP. You down at APP? That's amyloid protein precursor. And when this protein gets cleaved or chopped up by enzymes, first beta secretase and then gamma secretase, the byproduct is amyloid beta. So APP is the first step in the chain toward amyloid beta. So they started looking into amyloid, the APP, and trying to figure out, okay, where does APP come from? Well, the lab that was in charge of all this research, they looked at the brain's nerve cells, and in a study using mouse neurons, they were able to determine that there is another form of APP that's called PAL APP, but it's not friendly. It's not a friendly PAL. Mm-hmm. PAL APP means that this amyloid precursor protein went through a process called palmitoylation. Palmitoylation turned it into PAL APP. Then PAL APP gets transported on a raft of lipids. There are lipid rafts in our neurons that are called, they're mitochondria-associated endoplasmic reticulum membranes, MAMs, MAM rafts. These MAM rafts grab up the PAL APP and take it for a ride. And when the rafts take PAL APP for a ride, they go, oh, here's the beta secretase. And then the PAL APP gets all chopped up, and that makes amyloid beta. So in their study, they started blocking or upregulating the MAMs. They got rid of the rafts or they added more rafts, and they found that it totally increased or decreased the production of amyloid beta in these mouse neurons in a dish. So they were able to they were able to use a drug for a protein that's involved, the sigma-1 receptor. So there are drugs that could potentially be used to target a particular receptor that can control the rafts, that can control the production of amyloid beta and stop it from ever happening. So potential intervention for stopping Alzheimer's disease.
Yes, please.
We'll see where it goes.
This show has really made me optimistic about this in particular. I feel like, everyone talks about curing cancer in our time, and there's been interesting strides and stuff, but it really feels like Alzheimer's might get figured out in our lifetime. They're really getting close.
That's not saying much, Blair. Seeing as how...
Oh, because I'm going to live to be 200. I can't live to be 200.
Blair's going to live forever. That's right.
So it's like, wow, in your lifetime. Well, that's not
No,
really
in your lifetime.
Oh, well then, in that case, it better hurry up.
Well, anyway, I love this. I love the idea of everyone I know, and even people I don't know, not getting Alzheimer's. That would be great.
It would be great. Let's keep our memories, not just through memory palaces and storytelling in our brains associated with landscapes. We don't need to. We want to keep our brains working and our memories working at the cellular level as well. Yeah. So you down with APP? No way.
Mm-mm.
No way.
Nope. Get out.
I'm not down with APP.
You're not allowed on the raft, buddy.
Get off my raft. No rafts for you. All right. Have we done it?
We did it.
Oh my goodness, did we?
I think we made it to the end of another show. Thanks for listening to the show, everyone. I do hope that you enjoyed it. I want to thank my co-hosts for their amazing co-hosting. Thanks so much, Blair and Justin, for another good show. Thank you, Fada, for your help with social media and show notes. Thank you, Gord, for help in our main chat room. Thank you, Identity4, for recording the show. Thank you, Rachel, for all of your assistance. I also want to thank our chat room right now. Thanks for being here, everyone, wherever you are. And big thank you to all of our Patreon sponsors. Thank you, John Ratnaswamy, Kira Carl Kornfeld, Melanie Stegman, DeCrampster, Karen Tozzi, Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Shefstead, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shoe Brew, Darwin Hannon, Donald Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis104, Skyluke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Raleigh, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minnis, Johnny Gridley, Kevin Rails, Back Flying Out, Christopher Dreyer, Mark Mazzaros, Artyom, Greg Briggs, John Atwood. This profile name is hilarious in the context of some other podcast. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecorari, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you are interested in supporting us on Patreon, head over to TWIS.org and click on that Patreon link on next week's show.
We will be back.
Except Justin won't be.
That won't be. I'll be on an airplane somewhere over the Atlantic Ocean, probably.
Come on, they have Wi-Fi.
Broadcasting live, though, from YouTube and Facebook channels and from TWIS.org live. The show will continue.
Want to listen to us as a podcast? Maybe go for a nice run while you listen to science? Just search for This Week in Science where podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can sign up for a newsletter.
That's right. You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair at blairbaz@twist.org. Yes, all the domains are different. Just put just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into, let's say, bubble on the nose of an anole, because I didn't want to get gross.
Bubbles are nice.
I took the less obvious choice. All right, moving forward.
You can also ping us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you learned anything from this show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.